All right, thank you so much for my four wonderful panelists. Alex just gave a brief introduction, but to go a little bit deeper into each of their backgrounds, understand why they're qualified and experts about the science of meat. So we'll start with Dr. Kate Krueger, who is one of the founders and the current research director of New Harvest, a research institution that funds innovation in food engineering. She was also working with Perfect Day in their very early days, contributing to their foundational patents on novel milk proteins. And Kate has a Ph.D. in cell biology from Yale University. Next, we have Dr. Vítor Santo, who is director of Cell Ag at Just. He's previously worked with cell-based models and therapies for biotechnology and food applications, and he has a Ph.D. from the University of Minho in Portugal and the Kyoto University in Japan. Third, we have Dr. Nicholas Legendre, who is director of biological research at New Age Meats, has a research background in analytical chemistry and skeletal muscle regeneration. He has a PhD in molecular and cellular biology from the University of Connecticut. And finally, we have Dr. Eric Berg, who is professor of animal science at North Dakota State University. He is currently a fellow at the American Meat Science Association and on the board of directors. He's a PhD in meat science from Purdue University and a postdoctoral fellowship at Texas A&M University. And his research focuses on improving meat quality and understanding the nutritional and health benefits of meat. So today we're going to talk about the science of meat. What the heck is it? What is it made up of? What is the structure? And most importantly, how do we recreate that structure and quality in vitro? So to start out, we're going to talk about ground meat, so anything that's kind of in crumbles. So to start out, I wanted to ask Dr. Berg, what exactly is ground meat? What is kind of the structural components of it?
Yep. As a member of the American Meat Science Association, last year we were posed this question many times, what is meat? And we have a scientific journal called Meat and Muscle Biology, and published in that open access journal is a lexicon of meat science terms. And we brought together a panel of experts to put this lexicon together in response to questions that arose during the development of the dietary guidelines for Americans that they put together every 5 years. So right now they're working on the 2020 dietary guidelines. And with that expert committee, there were a lot of questions, Robert, about what is red meat? What is just meat in general? What's the difference between a processed meat and a whole muscle cut? So this lexicon was put together to help those regulators to identify that. So your question was, what is ground meat? We would classify that as a minimally processed muscle food. So at its minimal amount, a processed food in this case is just a ground whole muscle. And usually we associate it with something like hamburgers. But it's in many cases the first step before further processing where different ingredients are added, like spices or curing agents. And then there could be followed by different cooking techniques or even fermentation products to create different sausages or food products. So ground meat is basically taking a whole muscle and making it smaller.
Gotcha. So when you talk about minimal processing, that processing is basically just grounding it up, right? And so when you start with a whole cut that has a very complex structure, texture, what is maintained when you move to a ground product? Like, are the muscle fibers still intact? Do they clump up? Yeah, can you talk more about that?
Sure. So it depends on if it's a coarse ground so think in terms of bulk sausage, like you might have Italian sausage for pizza toppings. So that would be more of a coarse grind. So as you get smaller and smaller, it is going to expose more of the nutrients that are inside. So at the risk of this sounding kind of gross, it's like chewing. So as you chew, that's the first step of the digestive process. So by chewing, you're exposing the access to the nutrients, no matter what food you're chewing. It doesn't necessarily have to be meat. It could be a plant-based, anything. It's that chewing process makes a finer piece. So those nutrients are going to be released. And then if you think in terms of a hot dog or a bologna product, that is almost, we call it a batter even, because it has the same consistency of a cake batter. And at that point, you've emulsified both protein and fat. And at that point, the protein will actually encapsulate around the fat, and that gives the unique mouthfeel that you have in bologna and hot dogs. So with regard, if you get way down to sarcomeric proteins, actin and myosin, those proteins are going to remain intact. But if you apply a process, a cooking process or a fermentation process, then those nutrients are going to be exposed even more. So in plant-based foods, many times we ferment products to improve the body's access to the nutrients that are in those plant-based products.
Cool, that's very interesting. When we're thinking about, for example, a ground beef, think about something of the texture of the Impossible Crumble, if anybody's had that. Those chunks are a little bit bigger than maybe what you'd find in a hot dog. So in those cases, are the muscle fibers still intact, do you think? Or is it still kind of like, are you still getting this kind of encapsulation effect with the proteins?
Yeah, I think the muscle fibers would be intact for the most part. There's going to be some shearing that goes involved because there's a blade that cuts it. So there will be portions that are intact, yes.
Cool, so I think one really interesting difference between conventional meat and cultured meat is that when you make ground beef, you're starting with something bigger and making it smaller. But the opposite is kind of true when you're making it in vitro. You're starting with something small and trying to make it a little bit bigger. So, Dr. Legendre, maybe we can start with you. Can you describe the process of how you can get from something like free-floating cells in a bioreactor to something that we'd maybe recognize as ground meat?
Sure. I just want to start off by saying that skeletal muscle is a complex and heterogeneous tissue. You not only have the working cells, the myofibers, some of them which can be the largest cells in the body going from your hip to your knee. You also have vasculature. You also have a lot of connective tissue. In some cases, the dry weight of meat can be 1 to 10% collagen, which is a very abundant protein in the body and found a lot in a lot of connective tissue. Additionally, you have, I think a recent paper identified 10 mononuclear cell types that are their stem cell progenitors or vasculature or connective tissue cells like fibroblasts. So there is a lot of cells, a lot of things going on in whole muscle, right? Now, the vast majority is going to be skeletal muscle, the myofibers, the actual cells. But you're also going to have fat as well. And as you get an older animal, you tend to have a little more marbling from different progenitors in the cells. You have intramuscular fat. And to really recreate what you're going to get from a conventional livestock, from a cell-based, really takes a bit of work. New Age Meats had a tasting last September of pork sausages. They were able to take a biopsy from a pig, grow them up, mesenchymal stem cells, they were from the belly of a pig, and they differentiated them into muscle and fat. And they made 3 sausages, and there were about 15 to 20% meat each in those 100-gram sausages, and that took a lot of work, and that's obviously not feasible. But working with a sausage maker who used extenders like Rusk, they made English breakfast sausages that apparently had a very good mouthfeel and was very well received. This is before I joined the company. But in general, when you go from a bioreactor to something that's food, you're going to have to, in our case, grow up the cells separately, muscle and fat in different bioreactors, get to a certain density, differentiate them to a certain amount. Now, do you need all of them to be the long muscle fibers or the mature fat? I don't know off the top of my head. That's going to have to be empirically determined by a company. But you have to harvest them. You're going to have to get rid of any sort of contaminant, anything that may be in the media that you don't want on the cells, washing them several times. And our sausage maker, from what I understand, again, I wasn't there, he was able to take the the cells, they were spun down, and then he did his magic, so to speak. I'm not a food scientist, so I don't know exactly how he added it, but there were a lot of extenders, and it wasn't pure product on our end.
Dr. Santo, I know that Jess has also done a number of tastings. In your experience, in your experience, what is the process of going from kind of the individual cells in the bioreactor to something we'd recognize as meat? What is that like?
Yeah, so I think that a lot of Nicholas explained right now is the reality for most cultured meat companies here. So we have to take conventional meat as an inspiration and really understand which are the cell types that we're really focusing on culturing. I think all of us can agree that it's impossible to grow all the cells you will find in a conventional piece of muscle in a feasible way that is cost effective, at least nowadays, with the technology that we have. So I think that we have to be also realistic about that and focus on the main cellular components that conventional meat has. And obviously we need to look at the main groups like muscle cells, fat cells. There's also vasculature and the connective tissue. And you can grow them individually in bioreactors. You can establish co-cultures, right? So if you think about, let's say, the strategy we're describing right now for a more ground type of meat approach, and we grow them individually, then downstream of production, you can do some sort of concentration steps where you can assemble not only cells, but also some other ingredients and materials that will recreate some sort of, or mimic the structure of a ground product. And actually, I don't think this is that different from what happens in a lot of these ground meat products nowadays. If you look to the composition of, let's say, a patty burger or a sausage, there's a lot of also bulking and sort of binding agents that are essentially gluing together as well a lot of the conventional meat materials. So there's even some plant-based materials in there. There's cereals. So essentially we can follow very similar approaches that are already used nowadays for conventional meat products.
Gotcha, thank you. Dr. Krueger, so they were just talking about muscle and fat as being kind of the two most important cell types for getting the taste and texture of meat right. But in your opinion, do you think that there will be any more types of cells that companies will want to incorporate in the future? Maybe in the longer term, once initial products are out, is there some way that products will be deficient because they lack X type of tissue, in your opinion?
It's possible. I think it's really hard to say until we actually have the products to try. I tend to think that there's probably a lot of components of actual meat that we don't really need to have in meat products. So things like small quantities of different kinds of cells, as you mentioned, that might be, say, immune cells, et cetera, probably don't play a big role in much of the taste or mouthfeel. But it's quite possible that we might instead want to kind of augment what we can do with the current muscle cell types. So that's a reality I think is a little bit more promising, especially with rapid genetic modification techniques like CRISPR, where we could, in theory, engineer in additional properties, flavors, et cetera, to existing cell types. So I think rather than having a more narrow canvas, we may in fact have a broader canvas.
Yeah, that's something that's very exciting. Do you have anything off the top of your head that, in particular, you're excited about? Like, do you know of like a particular gene that would give meat a certain sort of quality that you're particularly excited about?
Yeah, so there are a couple. I mean, I think if you could add a whole biosynthetic process pathway for things like, for instance, the various B vitamins, for instance, or other classes of vitamins that are a little bit higher value and harder to get bioavailable, I think that would be a big plus. I think something that's dogged a lot of the supplement industry is the fact that while you can purify these compounds and such, they're not always the most bioavailable. So I think that is a big promising point for the field of cellular agriculture.
Cool. Yeah, and I'd love to drill deeper into that even. So I'm not familiar with how exactly B vitamins work, but is it just a matter of there being some gene or collection of genes that's responsible for cells being able to synthesize these compounds by themselves, that we can just, if we wanted to use genetic modification, just give to a cell? Is that what it is?
In theory, yeah. So, I mean, I think we're kind of talking in pretty broad strokes here, but basically, yes, it's a lot of one enzyme processes another enzyme that creates what you're looking for. So I think there are all sorts of these processes that we could add to augment various foods. Another pathway we could add in theory is something like lycopene. So if we wanted to, say, get more like tomato-like compounds, for instance, in different meats. So that's
what is lycopene?
That's the tomato compound that everyone talks about in ketchup. But that's just a random example. I think there's all sorts of interesting kind of high-value products that we could think of having in foods in the future.
So one thing I think it's hard to visualize for folks who aren't directly working with cells is when you have a culture in a bioreactor and you say proliferate the cells to a certain density, what does that look like? What is that material like right out of the bioreactor? Do you have like ground meat in the bioreactor, or is it something else? Anybody care to jump in there?
Yeah, I can jump into that. So, yeah, I mean, if you're talking about the culture of single cells or small clusters of cells, what you will see is a suspension that will look more and more dense and saturated, right, when it is in culture. And at the end of the process, if you concentrate those cells, it actually has a pretty similar structure and texture to a ground product. So it's not different than typical cell therapy approaches, for instance. So essentially, sometimes when you see the bioreactor, it's difficult to envision a meat product because essentially you just see a solution. But at the end, when you remove the culture media and you just concentrate the cells, it's actually, pretty similar. This is assuming, again, not using, for instance, a scaffolding approach, just using single cells and clusters or aggregates.
Suppose that you were using a scaffolding approach, how would that change things?
If you're using a scaffold, let's say for a more thick cut of meat, I would say that the bioreactor design is to change significantly. So you no longer use the conventional stir tank bioreactors, for instance, and you need to think about other type of bioreactor designs where you need a more efficient perfusion of the nutrients and oxygen transfer as well through the deep layers of that construct. So in that case, when you look at those constructs in culture, they will be much more similar to the final product that you're expecting from a thick construct, a thick piece of meat. So that's a parallel approach.
Cool. Now I think we can jump back to Dr. Berg. When in conventional meat production, obviously when an animal is slaughtered, there are a number of chemical processes that happen within the body. For example, ones that lead to rigor mortis, that sorts of things. And I imagine there's various ways in which these chemical processes can end up changing the end attributes of the meat. So could you talk a little bit about what happens post-slaughter inside an animal and how that might affect the taste, texture, or other qualities of the meat?
Sure. I want to follow up a little bit on what Nicholas said. Sure. With regard. I teach the muscle physiology class, and one of the first things I say is that everything that affects that living animal from before it's born, first during development and throughout its whole life, including the environment in which it's raised, the stress it's under, the food it eats, the amount of exercise it gets, all of that's going to affect this tissue we're talking about. So with that in mind, the conversion of living muscle to meat is very energy dependent. So think about how muscles work, just moving that animal around all its life. That requires energy, and the ultimate energetic currency is ATP. So in the process of stored fuels like glucose is glycogen, or fatty acids in adipose tissue, whether it's aerobic or anaerobic metabolism that's ultimately providing ATP, for myosin to hydrolyze ATP and bind actin to cause my leg to contract, my dog to run. You know, it all is going to impact that conversion. So now, if at slaughter, the animal is humanely stunned and it's the ultimate anaerobic condition. So now the circulatory system is gone, so that energy is an anaerobic system. So in a living system, we call that fatigue. So when you're working above your aerobic threshold, that burn you feel is glucose being metabolized and converting to lactic acid. But in a living system, that can go through the circulatory system when you're recovering, goes to the liver, and it can be converted back to glucose as gluconeogenesis, but it leads to fatigue. But that decline in pH in the muscle is the initial start to the conversion of muscle to meat. So we go from a living pH 7-ish down to something below 6, between 6 and 5, depending on the species. So that's the initial conversion. And rigor mortis occurs when that post-mortem accumulation of ATP is depleted, because you need ATP for myosin to release from actin so it can contract again. And if it's depleted, it stays, so you get that contraction. So again, everything that happens at that packing plant is going to impact the meat quality, because if this is a stressful environment, or you have an animal that's genetically prone to stress, just think about how you are when you're tense. You gotta go get a massage to work out the knots. If that happens in animals, we get an overcontraction, that's gonna result in tough meat. So the industry has gone, and part of my research is, I like to consider myself a holistic meat scientist, is to identify points of stress in the production system, to eliminate that stress so that it enhances the efficiency of muscle production and ultimately fat production, and how the energy is stored and how it's going to affect the ultimate product. So it's depletion of energy. And then many times you see steaks advertised as dry aged, or there's an aging process, especially in the beef or red meat species, that occurs. And then you start getting degradation of the sarcomere that starts to break down and improve the tenderness over time. So there's a whole series of proteases that are active early on, and then other things that occur during that aging process. So it's a very diverse system, as already been pointed out, very dynamic and extremely interesting to study. So all this stuff is very cool to look at.
So you'd say the quality of the muscle, which is partially a function of like how tense the animal is over the course of their life, then flows into the tenderness of the end product. Is that what you meant?
Yes, and when Kate was talking about there's some things maybe we could eliminate in a cell-based system, we don't need all that extra connective tissue that's found in the muscles of locomotion that are moving an animal all around all the time. That's why we grind that product up into hamburger, because it has that connective tissue to maintain the focus of that contraction. So these animals are prey species in nature, so those muscles need to be strong in case they need to run from a predator. So there's accumulation of connective tissue in there. The muscles we save as steaks are more muscles of posture, like the ribeye steaks or the tenderloin steaks that are on either side of the spinal column. So that's the big difference between a whole muscle cut and a ground cut.
So if you had to make a prediction, would you guess that cultured ground meat may be more tender than conventional ground meat, because you're not using these very thick parts of the animal?
If I were to form a hypothesis as a scientist, because in the absence of data, I can't make a conclusion, so my hypothesis would be that it would probably be more tender, because right now what we've been talking about is a ground product. So as this fascinating technology evolves, and where we get to a point where there maybe could be a whole muscle type thing, then we'd have to try that too to see how it is with regard to palatability aspects of tenderness, juiciness, and overall flavor.
Gotcha. So moving back into the in vitro side of things, the process of culturing cells and differentiating them, nowhere in there do we talk about the process of rigor mortis, this kind of change in pH. Do you think these sort of post-slaughter processes are important to replicate in vitro, or do you think that they are things that we can avoid because we're growing cells? Dr. Legendre, maybe we can start with you.
Yeah, that's a good question. You're going to have to look up what the processes do and not just treat it as a black box that you need to replicate. If the process does something that makes the meat more tender or more flavorable, then sure, replicate it, assuming that the conditions are the same, the cell types, the media, that if it mimics what's in the blood and so forth. But if not, then you don't need to do some cargo cult science. You just replicate things because that's the way it's done.
Good job. Do you mostly agree with that? Any opinions?
Yeah, I definitely agree. I think that we have to do this stepwise. I think obviously those quality traits, for me, come almost like a second level for these type of products. I think first we need to get a great understanding of how to build a cultured meat product from the beginning, from the cellular level, understanding which cells are relevant, and then we can really work on different types of maturation steps and how to improve the quality of what we have generated. But I think some of these processes can be certainly very appealing, actually, to improve the flavor profiles of the cell cultured meat as well. But for instance, this rigor mortis thing is definitely something that it's very difficult to mimic in vitro, at least at that level, right? We would need a very functional muscle that is contractible and that really shows that sort of exactly similar structure. And I think that's, again, something in order for us to achieve a very similar process. But it doesn't mean that we cannot replicate some of the steps of that aging and maturation step as well. But I think that we have to look at this stepwise.
That makes sense. One other process that just occurred to me is exercise. Obviously, exercise is one of the main things that we think of muscles as doing. Dr. Berg, could you explain what effect does exercise have on muscle tissue, and does that affect the end attributes of meat products?
Right. So we talk about exercise having the biggest effect on muscle food quality. So it depends on when it happens. And depending on where the animals are raised, so we'll stick with beef for a while. So if these beef are grazing for the first part of their life, and they're transitioning from being on grass in a large pasture into going into a feedlot where they're going to be on grain. So they're getting to a progressively smaller and smaller spot. So that exercise is going to be minimized based on the amount of space that they have to move around. Now, there are regulations with regard to how much space is there. And ultimately, that exercise is going to impact the development, the growth of their muscles. As Vítor said, that contractibility. And in a living system, it's exercise post-growth, okay? So you've got active growth that's going to occur that's different than post-maturity when they reach their peak. So that exercise is going to act on these satellite cells, these myogenic satellite cells, which are dormant nuclei in that muscle fiber. So when they come out of dormancy, they add more nuclei to that multinucleated muscle fiber to add more DNA to code for more protein. So exercise has an effect to increase the muscle size. But exercise has a negative impact on what we would be looking at consumers with regard to palatability. Because at some point, we want them to have that marbling in there. And that marbling is intramuscular triglyceride within the muscle that adds that fat flavor like you see in a prime rib. Okay? So Exercise is counterintuitive to that. And therefore, in livestock production, many times exercise is perceived as a stressor. And it doesn't necessarily have to be a distress. You can have a good stress too. So we go to the gym to exercise, hopefully as a eustress. But if you do it too much, it becomes a distress to your body. So all of that is in balance. Now, if we're doing this exercise directly before slaughter by inducing stress, and it may not be running, but it may be just so tense because of fear from a novel situation, that depletes the energy stores, and then it changes the ultimate quality. So we don't want that exercise to occur at the packing plant, at the end of the production phase.
So it sounds like exercise probably isn't one of the processes that companies would want to be worried about reproducing, because if anything, too much exercise in an animal would be negative for the end colony attributes. Is that right?
You need to stimulate the cells, though, for them to hypertrophy, correct?
Well, there's actually other ways. There's a couple of fusion proteins that are necessary and sufficient for satellite cells to merge with the muscles. So you could exercise, or you could use a transgenic system where you force expression of these proteins, then force these cells to merge with each other and hypertrophy that way in vitro.
Cool, thank you. So I think now I'd like to transition to talking more about whole cuts of meat, slab meat, which obviously is a bit farther in the future, so may venture into speculation even more so than we already have been. But Dr. Krueger, I'm wondering if you could tell me what are the main strategies that cultured meat companies could use to eventually create these whole cuts of meat?
Yeah, sure. So I think we've touched on a lot of strategies that could be used, both through exercise, non-exercise methods, and others. I think there's also a little bit of white space out there in terms of thinking about things that whole animals cannot do, but that we could do in vitro. So things like, for instance, merging products with, say, mushrooms or different kinds of vegetables like celery or spinach, for instance. So all of these materials have structural components that are often based of cellulose or related compounds, sometimes chitosan, for instance, which are can be really good materials for cells to potentially adhere to with some help. And so these may be additional ways of getting some structure, getting some fiber in food products that aren't really traditional.
One thing I think is hard to visualize, I've always found it very hard to visualize, is when we talk about scaffolding or hydrogels or any of these materials, what does it actually look like on a kind of cellular molecular level when cells are attaching to these substrates? Are they kind of just like globbing on, or are they, because muscle cells are fibers, are they wrapping around? What exactly does that look like?
So I think it depends on what type of cells you're talking about, whether or not you're talking about cells that have differentiated or cells that have not differentiated, whether or not you're talking about certain compounds that are bound to scaffolds that might help cells adhere, whether you're talking about various surfaces that cells might adhere to well. So I think it can look like a whole variety of things. In some cases, cells might look a little bit more stuck to a surface. In some ways, they might be a little more stuck to each other, and that depends a lot on the tech you use to interface with the scaffold material.
Gotcha. And Dr. Santo, you briefly talked before about these kind of bioreactors that might be used for these whole cuts of meat. When cells are adhering to these substrates, does that generally occur post or prior to differentiation? And yeah, what exactly does that look like?
Yeah, so typically, if a scaffold is a good substrate for the cells will definitely attach to it very strongly, and they will start to get more spread out through the surface. And for instance, if you're inducing muscle differentiation, with time, you'll see actually the formation of these myofibers and diffusion, right? So actually mimicking a very conventional muscle tissue as well. If a cell is not so amenable for attachment on those substrates, typically has more of a round shape, and it's probably not a good fit between the material and the cell itself. In terms of the differentiation of these cells in the scaffolds and incorporating that in the bioprocess. I mean, here, I think that we have to take into account two things. When a cell is differentiating, it's not proliferating. So also, as we need to achieve high cell densities to be, efficient in our process, it means that we have to either do two things. For instance, have a step where we just expand the cells, let's say in a conventional bioreactor, and then we have a second step where we do what we would be calling a maturation, so formation of a muscle tissue and induce a differentiation. That's a possible way, because otherwise it's just very inefficient. So you can produce the cells in bulk and then see them into a structure. For instance, it could be even in a downstream setting, outside of the same bioreactor where you grew the cells, and then have that sort of maturation happening and inducing the differentiation that can have an impact on flavor and also on the texture of the final product.
One thing I've, again, found hard to visualize is when you think about muscle fibers in a tissue, they're all pointed in the same direction, right? There's kind of these collections of, these bundles of fibers that are surrounded by connective tissue, and they're all going in the same direction. So is that something, when you have an in vitro scaffold or structure, do you also have some sort of directionality in that, or is it kind of more ad hoc with the muscles kind of like forming different directions? Does that make sense?
Yeah, I think that depends also on the scaffold design. So one good thing nowadays is that there's a lot of technology out there where we can have a very precise control over scaffold architecture in terms of porosity, percentage, interconnectivity. So obviously for a cell to be able to colonize, let's say, a scaffold, it needs to be highly porous, and these pores they need to be connected to each other. So there's diffusion of the nutrients, the media, and also the cells are able to sort of expand and go into the inner layers of this scaffold. So in the beginning, maybe 20 years ago, in the fields of tissue engineering and regenerative medicine, these structures were very random. So this porosity and interconnectivity, we didn't have much control over it. But nowadays, for instance, with 3D printing, we can very precisely define the geometry and the architecture of these scaffolds in a software, and then, for instance, just print the structures as we want. So that's an example of how can we actually try to align or to have a more defined alignment of the cells, mimicking a conventional muscle tissue.
Gotcha. So if you were just to say 3D print a scaffolding whose pores were basically aligned in a certain way, then the cells, when they are moving through those pores and attach, would eventually form myotubes in like the direction that the scaffolding was pointing. Is that right?
Yeah, that's correct.
That's pretty cool. Cool. I think that one thing I'd love to talk about while we have Dr. Berg here, because I think us four are all kind of from the cultured meat side of things, but you're coming from the kind of more conventional animal agriculture side of things. So I'd love to hear if you have any general thoughts, lessons from your work that you think the industry should be paying attention to, things we may be neglecting. If you could talk about that.
Yes. When we do taste panel studies and we do willingness to pay surveys and things like that, in many cases, what consumers, what customers say don't play out in the market case. And so that type of research and building a loyalty from consumers is probably something that the conventional meat industry has really focused a lot of effort on. So right now, we see a trend with consumers for clean label. Many of you with the plant-based proteins, they've received some criticism because of their long label. And the conventional meat industry has been working very hard to have a small label that includes ingredients that people can recognize. So I guess that's something that this new technology can learn from us, that the consumer loyalty has to be earned, because you have to have, first and foremost, a safe product, because people are concerned for their family safety. And next, it has to be a point of purchase that they can afford. And lastly, I would say that if consumers don't trust our technology, how will you convince them to trust your technology?
It's really interesting. On the product side, anything come to mind? Like, I think for a lot of companies, maybe they've done taste tests, but a lot of the product work is potentially kind of like theoretical. As someone who's worked with improving meat quality, does any lessons come to mind for how cultured meat companies can make sure that they're putting out the best products that they can?
It's easy. When you go to the restaurant and you take that first bite and you either go, wow, or you go, oh, that's the last time I'm going to buy that. Why didn't I go with my regular thing? It's the same thing as trial and error. To me, as an outsider looking in and having visited with many people in this industry, it seems like there may be ways, like Kate said, to enhance the taste and the flavor of these at the cellular level, which to me is very interesting. So if that can be done, there may be a way to more rapidly make changes in the organoleptic properties. Again, a hypothesis and an observation. So that's very interesting to me, the whole flavor science of this. And with aging of products, whether it's cheese or meat, it's the proteins forming salts of glutamate that are acted on taste buds for deliciousness, for umami. So identifying some of those things to enhance the deliciousness of the product. And again, it's marketing research, it's trial and error, it's real grassroots taste panel type stuff.
I wonder if any of the folks from the cultured meat side of things have found that there are lessons to be gleaned from the plant-based meat side of the alternative protein industry, just because they obviously think a lot about how to get all of these organoleptic properties right. Any inspiration there?
I'm not selling. Yeah, it's a tough question, Robert. Definitely, we've thought about it, and I know that New Harvest actually has a fellow that's trying to engineer more nutrients in meat. Andrew, right, Kate?
That's correct.
So yeah, we've definitely thought about it as a field, and as a company. Sometimes I think about things like, I know beef and meat there's very good sources of vitamins B12, which we kind of talked about earlier, and you're not going to get that from cultured meat unless we add it or have it to have the cells make it genetically modified cells. It's just, it's a tough challenge, and we are working on it, but I can't say too much more than that.
Dr. Krueger, I'm wondering if you could talk more about the project that Andrew is working on, if there's something more tangible that he's trying to engineer into the meat.
So first of all, I'd say definitely please take a look at our website. It's always great to kind of read what we've got there on all our different fellows, and we're really excited about the progress. So we do have one of our fellows who's working on some nutritional engineering for food products. So I definitely refer you to taking a look at that project description. But suffice it to say, he's working on some genetic modification strategies to add different compounds to meat that we wouldn't normally find there. So that's kind of a short explanation without going into the weeds, and I definitely encourage you to read up more on that.
In that sort of work, have you found that when you're genetically engineering cells to make these new compounds, I imagine would you control the cells to produce these compounds like in the proliferation phase, or is it like, when would the cells actually produce this compound? Like, would you end up with a lot of this compound in like free floating in the media when you're making the meat? Does that make sense?
So that's a great question. So that's one of the many things I kind of love about the world of cultured meat is it's all in your control how you decide to engineer these things, right? So some things may be easier to do than other things, some things may be cheaper, some things may be more expensive, but all of these strategies can be explored. So are you going to make some of these products intracellularly? Are you going to make sure they're expressed within the cell? Are you going to get them secreted or something that the cell kind of spits out into the media? I would bet that most of the meat applications are going to be best served by things that are made inside the cells instead of secreted, although different strategies could be applied. I think a topic that Dr. Berg just mentioned, which is the idea of genetic modifications for taste and flavor optimization, is really promising as well. I've not heard too much about work in that space going on just yet, but you could imagine things like increasing glutamine production, for instance, prior to harvest in a way that might give you an intense umami flavor, all sorts of possibilities in that kind of flavorscape or foodscape.
Is the reason that you foresee these modifications happening intercellularly, is that because you want to avoid interference of those compounds with the kind of culture conditions, like in the media, in the bioreactor and that sort of thing?
So depending on what people are growing and how, it would be my guess that in many cases, things in the media might, as Nina from Ospen mentioned yesterday, be recycled potentially or removed. And you may be separating the solid meat tissues from the media components. So I suspect it would be more kind of nutritionally complete to have things expressed intracellularly. But if you're going for a different strategy, where you're looking to maybe save high-value byproducts in addition to what's going on inside the cells, maybe as additional income stream, for instance, you might have different strategies around that.
Cool, that's all very exciting. I think now we should transition to audience questions. So I'm not actually sure how this works. Gotcha, cool.
Hi, I have a question that I'm not sure if it's science, maybe it's more engineering, but it's about scale. I'm wondering how big your production facilities could be, or you want them to be, because I know that they have to scale to an extent to bring the cost down, but I don't know how much. And I ask because I'm wondering if production facilities could be basically like in cities, so cities could produce their own cultured meat, or if it would be like there's one or two production facilities for the nation and then it's shipped around like we currently kind of do.
We definitely need a very large scale, especially if the mission, and I believe the mission of most cultured meat companies is really to change the way that industrialized meat production happens nowadays and to provide a feasible alternative. So we're talking about thousands of liters up to hundreds of thousands of liters. So some of these scales have been done before in the field of biopharmaceuticals or bacteria fermentation. Potentially we can try to go even higher. That's a challenging task. You can choose either to have multiple bioreactors of smaller scale, but I would say that nothing below 10,000. It needs to be above 10,000 liter to be considered a commercial scale. Anything below that, it's pilot. So, if they can be installed in cities, sure. I mean, that's one of the advantages. But at the same time, we have to consider that the cost challenges, these facilities would need to be also in cheaper land due to the scale. So certainly not San Francisco for sure.
I think if I can jump in there, I think another thing to keep in mind is that big part of the reason that meat production facilities in particular are located in rural areas is because it's most cost effective to be located close to the food that you end up feeding the animals, because transporting food tends to be more expensive than transporting animals just because of like the weight loss. So, yeah.
I have a question about cooking. So in conventional meat versus the cell-based meat, I imagine that some of the less common cell types and some of the extracellular matrix and all sorts of other molecules are going to be relevant for getting the right like physical transformations to happen upon heating and cooking. So I'm wondering how you think about that when you're planning and engineering for your cell-based meats. How do you make sure that there's, or maybe the better question is, what are the key components that you need to make sure are in there so that it cooks the way that conventional meat would?
Yeah, you can look at certain properties like water retention, emulsification, jellification, and compare it to conventional meat products. That's actually a nice way of also select for certain cells. I mean, if you are lucky enough to have, let's say, 20 great performers cell lines in terms of cell growth, and you come to the food science department, that's a great way of streamlining then the final product. So you can certainly look to certain physical chemical parameters, and based on these properties, then you can also decide which other components are you going to blend your final material with. And for that, food sciences has already developed for other applications the plant-based type of meats, for instance. And in our case at Just, we also have a lot of learnings from our plant-based egg, how to really recapitulate some of these textures and flavors, and how to make the correlation between the physical chemical properties and the final cooking of these products. So we're using all these learnings also as well for meat.
I'm going to be a little bit greedy with my question and throw out two, and it can be an either or a both. I'm curious about your response to questions of the safety of these products, and then I guess the second part is common and or dangerous misconceptions you hear about these products, and what your response would be to, I guess, address those.
Yeah, sure, we're always cognizant of the safety of our product. Like the keynote speaker said, we want to make sure we're delivering a safe product to the public. We don't want anything, if anything untoward or bad were to happen with the first product, that would really sully the industry as a whole. So we really need to make certain that everything is safe, and there's various toxicology studies you can do that have been well established in food science, using mice or what have you. And what was the second part of your question? Or maybe, yeah. Yeah, sure. A lot of people, again, like the keynote speakers said, a lot of people think they've had our product already, but they've had the Beyond Meat or the Impossible Burger, so there's a lot of confusion in the public about that for whatever reason. Another misconception, I guess, is GMOs. I personally think the industry would be very difficult or impossible without genetically modifying cells, and I know there is a lot of debate in the public about that, not just about plants, but also about animals too, like the Aqua Advantage salmon that was made in the late 80s and was only approved in the US, I want to say very recently, so that was decades. And very recently, these, again in animal agriculture, these hornless cattle, which I believe have a mutation that allow them to be born without horns, so they don't injure each other when they're in those stockyards or in the concentrated feedlots. So that's kind of an uphill battle. I don't know if Dr. Berg has anything else to add about that, particularly in the GMO side for animal agriculture.
No, not my area. So it would only be opinion, and if you want my opinion, you have to buy me a drink first.
Also, a common misconception is that some of people call this lab-grown meat, and it's true that it starts in a lab, but it leaves the lab pretty soon in the process as well. So this will be produced in any other large-scale facility that won't look that different from a lot of other fermentation processes that we have nowadays and that we consume without even thinking. So that's generally one of the biggest ones.
Yeah, hey guys. Quick question about the sheer volume of variables that are available across the whole line. So how do you, in terms of experiment design, how do you think about the number of variables that are available, the number of options of experiments that you could have? And we have like the modeling guys outside. How do you think about using data and what opportunities are there for good experiment design and choosing which pathway to go down?
Yeah, that's a good point. I mean, you can essentially spend decades researching this and never having a product in the market, right? So I think each company has to define what is the priority. To be honest, for Just, we have been discussing here a lot of quality traits, and that those are definitely our priority as well. But we cannot forget that this is to be an efficient and cost-effective process and scalable, right? And for us, definitely that's one of the biggest priorities, because it's the main challenge nowadays. We can have the most delicious piece of cultured meat that if it costs $1,000, no one will buy it, and this won't be a success. So our design of experiments is really targeted towards efficiency and scalability. And from the winners of those, then we look into quality traits and food functionality, but that's a second layer. So you really have to be really specific about the questions that you address, because otherwise you get lost. But again, that's also the beauty of having several companies in the field nowadays. I don't know exactly how many we have, but I would say like probably 40 or something worldwide nowadays. So I think that different companies can have different approaches, but definitely we'll be generating a lot of data, and we need to implement the right tools and experimental design. And I know that New Age Meats, you guys have a lot of that as well, right, in the automation.
Yes.
I think that we're out of time, right? Yeah, cool. Thank you all so much for your insights and your wisdom.
Thank you.